Hi, everyone. My name is Kathryn Sykes. On behalf of Calviri, I'd like to thank the organizers for the opportunity to talk to you about some new product ideas against cancer, all based on frameshift errors. Our mission is to end deaths from cancer. We want to provide affordable products worldwide, and we want to do this by mobilizing a new source of new antigens and some new array technologies. The premise is that this is going to require early detection of cancer and early treatment of cancer. We're going to attempt to find these neoantigens. We are finding these neoantigens by taking advantage of a key feature of all cancers. They make a lot of DNA and RNA mistakes. So here's an overview, a comparison between those two kinds of mistakes. At the DNA level, where most folks are looking for neoantigens, it's very infrequent. This is a genome. It has to be QC'd. It has to be made perfect as often as possible. It's highly disadvantageous to have mistakes in the genome. That's not the case for RNA, which is a temporary copy. So variants occur, and they occur predictably. And this splicing, whether it's microsatellite slippage, are occurring frequently and in all tumors, not just hot tumors. And because they're not substitutions from point mutations, but rather frame shifts, they are more immunogenic. And this is the source of new antigens that we are focusing on. Here's an example of a real neoantigen that we've been identifying in some cancer cells in a particular preclinical model. This is the gene structure. Normally, the exons are spliced together as they should, producing a normal wild type protein. However, if the tumor cell splices incorrectly, this exon fuses to exon 1 in a different frame, generating a frameshift peptide. So how do these RNA errors actually become immunogenic neoantigens? Here's a comparison of a normal cell to a cancer cell. They're both making DNA and RNA mistakes, but the cancer cell makes more of them. They both have a QC system, but the cancer cell is overwhelmed. Not all of them are degraded. They both have a protein unfolded response system, but the cancer cell is overwhelmed, so not all of these are degraded like they are in the normal cell, and they end up on the surface of MHC1 and MHC2. This also is true for antibodies. So here's a drawing of the whole process, but basically the frameshift antigen that's secreted by a tumor cell eventually activates a B cell and eventually is a huge amplification of antibodies that recognize that frameshift antigen. So we take advantage of all those antibodies and we screen them on these high density peptide arrays. We generate these by building peptides. Here is a representation of 3 features, each containing multiple copies of a single peptide sequence. And an in silico synthesis, we're using photolithography and masks, we're able to pinpoint which feature we want to add an amino acid to, and we do this 300 cycles to generate 400,000 peptides in situ. There are 15 mirrors representing all possible exon missplicing errors. We dice this silicon wafer into slides that allow us to put them into a cassette that basically create an ELISA workflow. We use ELISA reagents and secondaries to quantify and image the immune system. This is an overview of what I just said. Tumor cells generate the neoantigens from these RNA mistakes that are then translated. The activated B cell generates a lot of those antibodies that recognize those frameshift peptides. We screen all of the serum and the plasma and the blood antibodies on these arrays in which we have synthesized hundreds of thousands of possible frameshifted neoantigens. Turns out this exact same process and the exact same screening and data analysis can generate for us diagnostics and vaccines. And I wanted to show you a little bit about that. First, diagnostics. Here we have an analysis of selected peptides, 100 selected peptides from 5 different cancers. and their reactivity on these different cancer patients across the top here. And they're sorted so that all the most reactive in the breast cancers are here, those most reactive in the stomach are here. So you can see there are specific frameshift peptides that are diagnostic of each of these different cancers, all of the patients that have each of these cancer types. And here's just a brief summary of some proof of concept experiments where we're able to get very good accuracy in stage 1, even pre-stage 1. This is at least a year before diagnosis, and also some of the later stages. So now I wanted to spend more time on telling you about some of the vaccine development efforts that we've had, since that's what this Congress is all about. First, I wanted to show you, well, those frameshift peptides that we have an antibody response, so we know they're immunogenic, but are they protective? So here's a preclinical experiment using the biopsy injectable 4T1 breast cancer cell line to test whether or not there's any protection from these frameshift peptides. So here in red are the tumor volumes of mice that were injected with a set of frameshift peptides, and here's the mock. So there's a significant difference in the tumor volumes that is protective. Similarly, lifespan is increased in the frameshift vaccinated, actually post-immunization, so this would be therapeutically vaccinated mice. And most outstandingly, the frameshift inoculated mice had basically no metastasis. And since that's what kills us in cancer, we thought that was an important result. So here, back out of preclinical now into clinical, some human work. Here's exactly the same data I showed you, but I want to demonstrate to you by pointing out a few spots here that these can be used as therapeutic vaccines. So all of the breast cancer specific diagnostic peptides are candidates for potentially protective therapeutic vaccine. And these would be off the shelf, so they would be pre-made. So we call them our fast vaccines for frameshift antigen shared therapeutic. So these would be much faster for production and less expensive for production than the personalized cancer vaccines. So we did do a preclinical mouse study to determine whether or not there's any difference in the protection between a personal cancer vaccine and a fast vaccine. And we saw no difference. Here we have our fast vaccine in the red. It is certainly protective relative to the mock or these peptides that were not reactive in the cancer patients on our frameshift array. So now I'm switching to a dog clinical trial. So this is not preclinical. This is clinical. We're developing as a real dog product, real dog vaccine. So here our objective was to look for a pan-cancer vaccine, one that therefore, because it was in all cancers or most cancers, could be administered before cancer, so a preventative or prophylactic vaccine. So here's a set of peptides that were identified on the arrays for these 8 different dog cancers that are legended here. And as I hope you can see, there are some peptides, like this particular peptide is found both in breast cancer and in lung cancer. So we did an overlay, it's kind of Venn diagram determination of how many frameshift peptides it would take, and the answer was 127, to cover all of the patients, all of these dog patients in these 8 different cancers. So we applied this concept to designing the vaccine composition for our VAX trial. So this is the world's largest dog clinical trial. We have enrolled 400 of the 800 anticipated full trial. We're 1.5 years into our 5 years, and we are working very, very closely with our 3 clinical research sites here at Colorado State, UC Davis, and the University of Wisconsin. Here's our website for our progress to date, and we're very excited about the possibility of ending cancers, possibly even by preventing it from ever starting. So in summary, I hope to have conveyed to you that we've unleashed a rich source of frameshift peptide neoantigens, aka RNA-derived errors, that Calviri can manufacture these frameshift peptides in a manner that lets us screen patients for which ones they produce, either for a diagnostic or for a therapeutic or for a preventative vaccine. And these are our unique products that we hope we have convinced you will be useful in the future. Thank you very much for your time, and have a good day.